First of all, I would like to thank the organization for this opportunity to present my work today, which is called the Reciprocal Regulation of Cell Cycle and Sphingolipid Metabolism. So, first of all, sphingolipids are these amazing molecules that can form biological membranes and also work as signaling molecules. And especially for today, it was shown that for mammalian cells that some species of sphingolipids can act to lead the cell cycle progression, as you can see here for sphingosine 1-phosphate and also ceramides. But in this work, we will explore if the sphingolipid metabolism can be controlled during the cell cycle progression. For this purpose, we use the Saccharomyces cerevisiae as a model, in which both processes are highly homologous to mammalian cells, and also the tools to study both sphingolipid metabolism and the cell cycle are largely described. And we are going to focus today on the first checkpoint of the cell cycle that happens during the G1S transition. And this checkpoint is characterized by a transcription wave that we can observe once cells are synchronized at G1 stage of the cell cycle, as you can see here at time 0. And the peak of those regulated genes occurs after 30 minutes of growth, and they return to basal levels after 60 minutes of growth, corresponding respectively to cells entering at S phase of the cell cycle and G2 end stage of the cell cycle at 60 minutes. And this checkpoint is regulated by two different complexes, the SBF and the MBF. They share a semi-regulatory subunit, the SWI6, and they have their own transcription factors, the SWI4 for the SBF, which is the homology of the mammalia E2F1, and the MBF1 transcription factor for the MBF complex. So first, we wanted to know if those two complexes were somehow involved in sphingolipid synthesis. We then treated the cells with myriocin, that with a sublethal concentration of myriocin that inhibits the first step of the sphingolipid synthesis. And we expect that cells with lower rates of sphingolipid synthesis are more sensitive to myriocin, as you can see here for this mutant. That is a mutant that lacks the subunit of the serine palmitoyltransferase complex. And we see that this mutant has a severe growth defect on the myriocin treatment. So we compare the sensitivity to myriocin of the deletion strains of those G1S regulators to the wild type. And we saw that the SBF mutant, that the cells that lack SWI4 transcription factors, are more sensitive to myriocin when we compare to wild type. And the SWI6 mutant and the MBP1 mutant, those mutants present a similar sensitivity when we compare to the wild type. So suggesting that the SBF complex and specifically the SWI4 transcription factor has a specific role in regulating the sphingolipid synthesis. And the effect of myriocin in all cases was reversed by addition of phytosphingosine, as we can see here, that cells can grow again. So our first hypothesis in that way was if SWI4 can regulate the transcription of the sphingolipid genes. And to answer that question, we use a database called Yeast, and Yeast can correlate transcription factors with target genes and also target genes to transcription factors. And they perform that analysis by using binding evidence, data sets, expression evidence, and also by doing the analysis of the promoted region. So we are seeing here the sphingolipid pathway in yeast, and in blue are the genes that are possibly regulated by SWI4. And one important thing is that SWI4 is not just able to regulate one group of sphingolipids, but several species ranging from sphingoi bases, ceramides, and also complex ceramides, as we can see here. We then analyze the transcription of each gene during the cell cycle progression, And we found two groups of results that I will present briefly here. The first group of genes are the genes that didn't present a transcription wave that we can see here in the black lines for the Y-type strand. This is an example for this TS10 gene that is responsible for the synthesis of the hydrosphingosine. We can see that the gene does not have a transcription wave. And also another group of genes that present this transcription wave, as you can see here for the SUR2 gene and LAC1 gene responsible for the phytosphingosine and ceramide synthesis. In both groups, we saw that deletion of SUI4 that is represented by those red lines. The deletion of SUI4 reduced the transcriptional of sphingolipid genes. So next, we wanted to know if SUI4 can also regulate the sphingolipid levels. We then grew cells for 30 minutes. That is the specific time that the SPF complex is active, and also the cells are entering at S phase of the cell cycle. We then harvest those cells for the sphingolipid measurements. And again, we saw that deletion of SUI4 reduced the levels of the hydrosphingosine, phytosphingosine, and also total ceramide levels. So this data is in agreement with our mRNA analysis. And so up to now, we saw that SUI4 is able to regulate both gene transcription and also the sphingolipid levels. But this upregulation during the G1S can regulate further stages of the cell cycle. We have some clues about how this is actually happening in the cell. But first, we are seeing here late stationary phase cells that are arrested at the G1 stage of cell cycle, as we are seeing here. And this is happening because the nutrients in the media are low. And so the cells reduce their division rates and then become arrested at G1. We then inoculate those cells in fresh media and treated them with myriocin. And after 5 hours of growth, those cultures reached the exponential phase, as you can see here. And we can't see much difference between control and myriocin treated cells. But after 1 day, we can see here for the control that cells are becoming arrested again at G1 once the nutrients are becoming low. But this is not happening for the myriocin treated cells. As you can observe here, cells treated with myriocin become arrested at G2 end stage of the cell cycle. And this effect of myriocin is reversed if we treated cells also with phytosphingosine. As you can see here, this is the same experiment. So we are seeing here the cells treated with myriocin that are arrested at G2 end stage of cell cycle. And we can see that cells treated with phytosphingosine have more, we can see more cells that are at G1 stage of cell cycle. So those results suggest that it's the lack of sphingolipids that arrest cells at G2M stage of the cell cycle. So we propose that during the G1S transition, SPF complex activates the sphingolipid gene transcription in a cell cycle dependent way or in a cell cycle independent way. And those molecules can signal for the proper cell cycle promotion. As you can see here, that sphingolipids can promote G2M progression, so at further stages of the cell cycle. So I want to thank everybody for their attention today, and especially to my advisor, Monica Lomeli, and everybody in the lab for helping me to do this work, and also the funding agencies for the financial support. So thank you,